to engage with our international collaborators and other collaborators. And today we're very lucky to have 4 of them. So we have Isla Meyer-Smith, who will give a talk, Phil Townsend, Christelle Schaff, and Pablo Arroyo from NRC. And so the program this morning goes like this. It's a little bit, we're a little bit behind, sorry, but so the first speaker will be Phil, and then we have Pablo from NRC, Isla, and then finally we'll have Christelle. There's the coffee break after, so you're all, of course, free to stay and interact with the speakers and other people. So we planned 25 minutes for each talk, and so Carol will be waving at 20 minutes yellow sign, and then at 23 minutes she'll be waving the red one. Great. So we'll start with Phil. I'll just give a brief introduction, and sorry, my notes are on my phone. It's not that I'm texting. So Phil Townsend is professor of forest and wildlife ecology at University of Wisconsin-Madison. Forest remote sensing, spatial analysis is his expertise. He uses remote sensing to study the functioning of ecosystems. He's published well over 100 papers. He's developed interesting outputs for the remote sensing community, ecosystem spectral information system, as well as SpecDAL, a Python library for preparing and processing field spectroscopy data. He's an affiliate at NASA's Jet Propulsion, JPL laboratory. And he works very closely with the National Ecological Observatory Network, NEON, and I believe this is what he'll be talking about. I'll be setting your talk. Just give me a second. Wait.
I wasn't quite sure who the audience would be, so I put together sort of a general presentation, and you're definitely going to have to stop me after 25 minutes, but a fair amount.
Thank you very much.
Take the microphone.
Yes.
Yeah. A fair amount of this will be sort of repeats of things that I think just about everybody here knows. There we go. So thank you very much. So I am going to talk about NEON. I'm going to talk about sort of functional diversity in vegetation through the lens of foliar functional traits, which I think a lot of you are familiar with. There are just a number of different traits. What are these traits? There are a number of different traits at the leaf level that can be scaled up to the canopy level that we're interested in. So we're really talking about function at the leaf level, but that's not necessarily what we're mapping using the NEON data. And we break these traits down into different categories of function that we're interested in with vegetation. In particular, the functions associated with photosynthesis. Some of these traits which can be detected using remote sensing, but there are a number of different functional groups of that we're trying to look at in addition to photosynthesis, so nitrogen, leaf mass per area, chlorophyll, sugars and starches, the outcomes of photosynthesis, for example, and then other macronutrients that are associated with vegetation performance. But there are other components of leaves and of canopies that we can see that are related to function. So structural compounds, things like lignin and cellulose that are very strongly related to decomposition, so the recycling of nutrients that vegetation depends on, and then defensive compounds such as tannins and phenolics. And the whole concept behind this was expressed in the Yates et al. Paper from 2016, in which they show that there are, when you look on the x-axis here, you see latitude, so from south to north, and then you see number of species that are in databases, shown right here, versus, right here, number of species that are present versus number of species that we have trait information, and there's this huge gap in information that we don't know. And so remote sensing, these are places we can't get to. Your field scientist can't be at all of these places. The data are expensive to collect and so forth. The notion is then that if we look at spectra at a leaf level, as shown here, there are lots of characteristic wavelengths at which there are absorption features that are associated with these different biochemistries. Some of them are very distinctive, single locations, such as chlorophyll and phenolics, for example. Others are spread across the spectrum, but the idea is that there's so much information content here that we can leverage and use with imaging data, shown here from NEON. This is from Florida, Ordway-Swisher Reserve in near Orlando, Florida, which we can then use to perhaps estimate some of these traits. We can't map all. You gave a pretty precise number of species that were there. Significant, I'm not sure about the significant digits there, but we can't map all of the species that are there, but we can probably map variation in functional traits that will allow us to get at some measure of diversity and some measure of the function of ecosystems. So this use of imaging, spectroscopy, and other types of remote sensing to get at vegetation traits is not at all new. The first paper that showed the use of imaging spectroscopy was by Westman et al. In 1988, with data collected on by the Airborne AIS spectrometer from NASA in 1984. This was a group out of the University of Wisconsin. Interestingly enough, that was looked at mapping lignin concentration and nitrogen concentration from imaging spectroscopy, with the idea of getting at decomposition and recycling of nutrients in ecosystems. So immediately there was this idea that we could use this information to actually estimate properties of ecosystem that we care about. And this led to additional work in the subsequent decades to see if we could make this more generalizable. There was a paper by Martin et al. From 2008 in which they collected data from multiple sites, mostly in North America, but also a site in Australia and a site in Costa Rica, to develop a generalized model using Hyperion and Avarice data to be able to predict nutrients, and then work that came out of my lab developing a generalized model for forests to predict a whole bunch of different traits: leaf mass per area, nitrogen, carbon, cellulose, lignin, and fiber, as well as isotopic N15. And not only map this across a lot of different sites, different years, different types of conditions, but also that second column on each side to map our uncertainty in our ability to predict this. So we're actually getting to the point now in terms of looking at traits to maybe use this information to start to fill in the gaps. And something like Cabo or NEON or other efforts would be looking at getting at that. And similarly, insofar as we can use this information to extend data, such as from flux towers or other types of measurements, we start to fill these data gaps. This is showing where the GPP is occurring on the planet. This is showing where the flux towers are. These huge gaps are really important, and to understand the context of climate change, we need to be able to fill this in, and we're not going to be able to put flux towers everywhere. So NEON, I hope that many of you are familiar with this. This is a U.S. project that was that broke the country up into ecological zones. These biomes, if you will, are perhaps a little bit more politically defined than ecologically defined, but the concept is that within each one of these, there are sites that are representative of the diversity within these biological zones, in which a lot of data are collected every year. And these pins from Google Earth are actually showing you where some of the sites are, but within each one of those sites, a lot of data are collected. The key data set with respect to us is the AOP, which is the Airborne Observatory, Airborne Observation Platform, which is an Avarice Next Generation type instrument that's flown on a Twin Otter, as well as high resolution photography and waveform LiDAR. And each one of these sites was planned to be flown, well, originally they were planned to be flown 2 or 3 times per year. Each one of the sites is actually flown once per year, and there's an elaborate process to identify the period of peak greenness when they're flown. In reality, about 80% of the sites are actually flown each year due to scheduling and weather and so forth. And so our project is actually to develop foliar functional trait maps for all of these. So it's not just forests, it's all ecosystems, and basically it includes anything that's flown within these airborne imagery boxes. So our area of study here is everywhere. Our functional traits are whatever we think we can get. Many of the functional traits, I didn't really go into this, many of the functional traits, there is a physical basis behind being able to estimate it. Many of them there is not. It's correlational, but we, well, I can discuss this more with folks later, but validation and calibration is really a key part of this. So we have spent 3 years now chasing the airplanes around the country and collecting field data. At this point, we've got over 1,275 field plots across most of these domains. The only one that we haven't sampled is the desert Southwest, actually. And in addition, NEON collects a small number of samples in each one of these domains when the plane flies as well. But they're sampling, we're trying to capture the diversity of vegetation, whereas NEON really focuses their sampling on the dominant vegetation within each one of these sites. So this is where we are working. And so the AOP data, this is from Talladega National Forest in Alabama, which is actually a really, really cool site. So there's the AOP data. This is the main thing that we're working with. It's imaging spectroscopy data. I think a lot of you have probably seen these imaging cubes of the data that we're trying to turn into maps of foliar functional traits. These are the traits that we're working with. I'm not going to go into the details. We use two different approaches for mapping. One is partial least squares regression. We use that because it's fast and it has a long history of being repeatable, but we've also used a more newer technique called Gaussian processes modeling, which actually gives about the same results, but it actually has a better way of dealing with uncertainties in the mapping. And actually there are more traits than this that we're working on. These are the ones that look really quite good. And I also haven't gone through the processing of the data. Etienne mentioned EcoSys, which is our spectral library, and I sure hope Cabo puts its data on EcoSys. We've also created this toolkit called HiTools, which is also on GitHub, can be downloaded, and it's pre-processing for airborne imaging spectroscopy data. So it's got a whole set of open source tools for BRDF correction, topographic correction, and other data handling needs that are unique to airborne data. And it works with NEON data, NASA data, and HiSpecs, the HiSpecs imaging spectrometer from Norway. The key point here is that this is actually the validation R-squares and the percent RMSE, and I show the percent RMSE there because that's actually probably the more useful metric than R-squared. NASA has set 20% as being the uncertainty that they're willing to accept with trait estimates from a forthcoming future spaceborne imaging spectrometer, such as the Surface Biology and Geology mission. And this is completely independent data that's used for this validation here. And one thing that we're showing is with our traits that our uncertainty rates are by and large, for the most part, less than 15%. And this is applied, these are models that are applied to all of the NEON domains and all vegetation types. When we separate this out into different physiognomic categories like trees, forbs or grasslands or something like that, we actually get much, much better models. But what we're trying to do is come up with sort of a blanket method to be able to do the predictions. So what I'm showing you right here is actually the results I'm going to show you are going to be based on a single model rather than models tuned to particular vegetation types. So this is just an example of the output of it, showing data from 5 of the different domains. This is leaf mass per area maps. This is just a true color image on the left, and then the map of leaf mass per area. LMA is a widely used surrogate for photosynthetic potential. Thicker leaves, slower photosynthesis, thinner leaves, faster. And then a lot of different traits using this Talladega National Forest area just as an example. So these are the sort of maps that we're developing. We've automated this, and we're working with a lot of imagery, so we work on a distributed computing system. So how do we visualize the trait variation? So this is what we really want to get at. The actual mapping is interesting, it's fun, I'm a remote sensing person, I like doing that, but it's actually the ecology that we want to do. And so a lot of people put this in the context of the leaf economic spectrum. I just sort of mentioned this from sort of slow processing with high LMA to low LMA, low nitrogen to high nitrogen, leaf longevity, gas exchange rates, and so forth. This has been widely used as a basis for understanding trait variation, and the Wright et al. Paper is kind of the most famous example of it. This data has been reanalyzed quite a number of times. So if we look at this one here, we've got LMA on this axis here. So photosynthesis declines as LMA increases, and there's a similar relationship with nitrogen content. So what I'm going to do is these data from the Wright et al. Paper, the original leaf economic spectrum data, I'm actually going to superimpose on our data from NEON sites a little bit later. How this links up to remote sensing is something that a lot of people want to do, and Asner et al. From his work in Peru a couple years ago in PNAS showed some of these relationships that were shown as part of the leaf economic spectrum. His results don't necessarily match what the leaf economic spectrum results show, which is interesting. He doesn't really comment on it in the paper, but there are some interesting consequences of these relationships that he shows, and some of the ones that I'll show in our results as well. So this work kind of started out from work that we did in Cedar Creek, LTER, and then forest sites across the U.S. And trying to visualize all of these different traits and what they mean is a hard thing to do. So one of the things that we do with the data is we'll do a principal components analysis of or principal coordinates analysis of all of the different traits. In this case, these triangles here in both of the triangles in both of these are imaging spectroscopy estimates of traits shown in principal coordinate space. So this is covering about 65% of the variation in grasslands. This is covering 86% of the variation in forest. And we can look at what the drivers are based on our leaf level estimates. These other colorful points here are actually individual species projected into the same PCO space. And the interesting thing is that the dimensions of variability, like increasing nitrogen, increasing LMA this way, match the leaf economic spectrum. But what's really become cool to all of us and what we want to do is to ask the question, is there unsampled space in trait space? And I don't know the answer to that. Are we finding trait characteristics that we're not sampling because as ecologists out in the field, we have biases, we sample certain types of things, we don't sample other things? Or is this an inherent quality of mixing of stuff within pixels in remote sensing data? I actually really like to look at the maps. And so this is that Talladega site. Again, we can look at the key photosynthetic variables here, LMA, chlorophyll, and nitrogen in the red, green, and blue. And we can see the conifers here showing up in the red and the broadleaf species showing up in blue. But it's really, I think, really interesting when we look at some of these other traits that we're able to map. So this is LMA, potassium, and phenolics, a defensive compound. So things that are blue are higher phenolics vegetation, and some of it is in the conifers. Conifers do have some significant phenolic. These are still the conifers that are over here. Some higher potassium species showing up here. Kind of interesting. We can start to ask questions about what the variation is and what might actually be important. I showed these three because they all had pretty good performance in our spectral models. Okay, so if we start to look at trait space, we can look at how samples from an individual site, shown in the orange, relate to the overall distribution of traits that we would see across all of the domains. And that's what's shown. So we can actually start to look at our functional traits distributed across within a site, distributed similarly as they are across all sites, and how much variation is there within a site. So this is just showing one site, Cirque, which is a very tall, beautiful coastal plain forest in Maryland. Once again, we can also start to overlay this with actual traits that drive this variation. And I'll just point that in this case, in the 2 dimensions we do see this photosynthetic dimension, so higher nitrogen and phosphorus versus higher LMA. So this is sort of the photosynthetic capacity, and this would be a dimension that would be associated with defense. And it's just showing two dimensions. We can look at how different physiognomic types fill out the domains, and again, we're starting to see, this is all domains now, we're actually starting to see unsampled space over here, which is really interesting. And I was hoping to show a map of the unsampled space, but we're not quite there yet. And we can look at how actual different domains, so these are the different domains that are shown on this particular figure. We can actually look at how different domains fill out the space. And you'll see that this is the standard deviation of each one of them. And you can see that they actually take up quite a bit of the functional space. But I'll actually point you to a couple of the domains. This gray one, which is again the one in the southern US that includes Talladega in Alabama, you can actually see that the field samples right here and the image data are not necessarily very close to each other. And that's, I'll skip this one because I think I'm probably running low on time, but I've got another graph that addresses that. We can actually look at how the different domains differ from each other. So the one that we were just looking at here was this domain 8. But this is actually what I was going to get to next. Does the field data and the image data show the same thing? So one way of characterizing functional diversity would be using something like convex hull volume. It just illustrates convex hull volume idea. And so we can calculate the convex hull volume and how it differs between the field data. Now, the first thing you'll notice is that the x-axis is different on each of these, and that's because the convex hull volume from the imagery is much larger than the convex hull volume from the field data because of what I showed you before. It just shows a much larger space, a lot more samples, and so forth. But what we'll see is that the ordering of these data aren't necessarily the same. You would expect that if our field data and our image data matched each other, that we would see the domains with the higher convex hull volume. And this is, by the way, this is the one in Alabama that I showed you. But we'll see here, for example, that our field data show higher convex hull volume with domain 6, which is where Kanza Prairie is. This is a prairie domain compared to the image data. So this is, we're just getting to the point where we have these maps and we can start to ask these questions. We haven't answered the questions why yet. We can also look at the data now in a lot more of the way that we would with the leaf economic spectrum. So this is showing the relationship between nitrogen and LMA. Right here, this is a very typical relationship. These are field data, and then the pink swath underneath it is the image data. We can look at nitrogen phosphorus relationship. I point this out because our data here with the nitrogen phosphorus relationship look a lot more like what the leaf economic spectrum would predict. This was actually not what the Asner diagram predicted. And then this graph right here is actually showing our data versus the GlopNet data that were used in the leaf economic spectrum, but this is actually only showing our field data here. So our data are matching, based on field data, what we would expect to see. If we then go and actually throw the image data on, so this is that same figure right here as the last one, but now we've got the image data shown there, we actually see something a little bit different, which Asner did also in his paper, and that is that if we blow up this image data, this right here is actually this here broken out by the different domains. We actually see a much more linear relationship than the inverse relationship that's shown by the leaf economic spectrum. And that's a really interesting question. So basically, function as leaf mass per area increases, you start to see photosynthetic capacity drop quite substantially. So this is the area that we're starting to go with this data, is to understand what the remote sensing has a lot more data than we can possibly have in these trait databases. So we can start to ask questions, what is the true relationship? So I'm just going to skip ahead and just show a couple slides from my student, Adam Kloos. Traits vary within a canopy. The traits that we're showing are basically the top of the canopy traits. But if we have LiDAR data or forest structural data, we might actually be able to estimate the variation in traits through the depth of the canopy. And that's something that Adam has been working on. So you take the imaging spectroscopy data, you get something about light transmittance going through the canopy from the LiDAR data, and you might be able to estimate the trait variation through the canopy. And this is his results. This is height by LMA, and then this is LiDAR transmittance by LMA. And you find that when you use a LiDAR transmittance estimated from the concurrent LiDAR data, you get a very good estimate, actually regardless of what the species are that are present. So this is actually all species in his sampling using LiDAR transmittance and LMA, which then, as you take an imagery, gives you the opportunity to estimate by height the LMA variation based on the different canopies. So from an aspen to a maple, and back to an aspen forest. And so I work mostly with imaging spectroscopy data, but I'm a really big believer that to fully understand ecosystem function, we need all of these different data types in addition to thermal and measurements on the ground and so forth. Okay.
Thank you very much, Phil. It's great to see this, especially that you're certainly a few years ahead of us in terms of trait mapping, and so it's great, very inspirational. We have time for maybe just a couple questions, and we'll move on. Remember that there will be a coffee break and time to talk to speakers afterwards. Maybe just a couple. Mark?
So a couple of times you mentioned that the remote sensing data gives you a sort of broader range, some spots in trait space that might be undersampled. And you gave a couple of reasons. One might be sort of the artifact that like a pixel has multiple things in it, so it's not really, there isn't really any detail.
Yeah, so the leaf measurements are done on leaves, and the pixel is seeing lots of leaves.
But
yes.
Later, you referred to the remote sensing data as giving us the true relationship that we didn't, and so you kind of like dismissed the hypothesis.
Oh, so I don't know what the answer is, actually. So next year, our sampling associated with neon is actually going to using the maps that we've now developed and sampling in the places that seem to be suggested, suggesting that they're outside of that trait space. And we're going to use that as a basis for sampling. And my suspicion is we're going to end up in places that we didn't expect, that we might not have sampled previously. I don't know what the answer is. I would suspect that it's the difference between a leaf economic spectrum, which is that relationship that we would expect to see, and a canopy or a pixel level spectrum, where the mix of what's present in a place might lead to more of a linear relationship as we have multiple species mixed in a pixel or multiple canopies. But I actually don't know. It could be completely plausible that we have undersampled the data. And really quickly, I'll just show one thing.
Sorry.
Oh, no, it doesn't matter. There are lots of pixels, and it obscures the fact that if you look at the density of where the pixels are, it's a little bit closer to that log-log relationship that you would expect to see. But it's a hypothesis. It's cool. I would be interested in hearing people's different thoughts on it, because those plots that we show of it being a linear relationship between nitrogen and LMA is not what the leaf economic spectrum shows.
Thanks very much, Phil. Sorry for the, just with time, we'll move on, but thanks again very much.
Thank you. Just make sure it's on. The pointer doesn't work on the screen, but you can use it to flip slides. So this one? Yeah.
All right, so the next speaker is Pablo Arroyo. Pablo is a research officer at the National Research Council Flight Research Laboratory. He was previously the director of the Geographic Information Center at McGill University. His research focuses on environmental applications of hyperspectral remote sensing, and he's worked for a number of years with Margaret and other people, obviously from NRC, that are here to develop the platforms that we use for our imagery, the UAV, but also the aircraft. And today he'll be talking about the UAV. Thanks.
Well, thanks, Etienne, for the introduction. Thanks, everybody, for being here, and the people that are online. I'm going to present this platform that we've been working and developing for the last 2 years. And although my name is there, this involves a lot of people, and Margaret here as a co-investigator, and Margaret students at an NRC. We have a big group of researchers that we've been working developing the UAV micro CASI platform. This is the outline of my presentation. I'm going to introduce the rationale behind UAV applications in environment, specifically forestry and peatlands, hyperspectral remote sensing, just a crash 101 in what is hyperspectral and why do we care. Although Phil did an excellent job already in show how we use it, kind of hyperspectral on asteroids, but I will be back a little bit and explain what is the rationale behind it, then describe the UAV microcasi system and some examples actually from Cabo, from this year first hyperspectral UAV measurements. Some of you might be familiar with DJI line of products that goes back 5-6 years. I'm not promoting DJI products here, but is one of the major vendors in the market, and when there is a new development, they have a lot of people working on it. They're very stable platforms, and we've been testing some of these platforms in collaboration with Margaret in their use for mapping forest structure, basically, and also peatlands mapping. An important part of your UAV is not only your platform, your rotocopter, or your fixed wing, is also your sensor. And that is fundamental. A good camera, expensive, is going to give you amazing imagery. A cheap camera is going to give you so-so imagery. So when you have a product, a map of that ecosystem, that information you can extract is limited. And again, in academia, funding is not flowing like millions and millions and millions of dollars, so we can buy all the toys possible out there. So we need to find compromises in what are the best tools available, right? So right now, I remember when I started, Margaret had the idea 8 years ago to start using UAVs, and we got a gas helicopter. And we started using that, and at that point, nobody was using UAVs mainstream, and it was fun. But again, it wasn't mainstream. Now we have these systems that are very useful. When we go to fixed wings, there are examples here, not only rotocopters, but I want you to get out of this is our ground samplings. Remember 6 years ago when GPSs give you 6 meters error and you were happy? Remember last year when you start getting 1 meter error in your GPS and you were extremely happy. Now we are talking about centimeter mapping, and that meter error doesn't make you happy anymore. So those are some of the considerations we are starting integrating into our mapping systems. And especially when we talk about forest structure, whether you use LiDAR or images derived from our drones through structure from motion to create those maps, or we use a species hyperspectral, for example, to map species richness, functional traits, and so on. This is a quick search I did a few weeks ago to present in Forest Sat conference in Maryland a couple of weeks ago. So I did just UAV studies for forest structure published since 2011. And you see that increase already. People are using these platforms for forest structure mapping. If you go document type and discipline, well, most of them are articles, peer-reviewed articles. When you go into the discipline, you will notice that we have remote sensing and photogrammetry journals publishing most of them, and some in forestry and then other fields. For me, that indicates that the technology is still under development. We are testing, we are trying to understand what can we achieve with this technology, but the application is coming a little bit behind. In forestry, do the forester feel 100% comfortable that those images or those products are totally useful and publishable? I think we have a gap there that is going to be probably filled in the next 3 to 5 years when there is complete absorption of the technology. Just two concepts here that are very common. I'm going to be talking about them. LiDAR is a very, at least in my opinion, Crystal, you might correct me, but it's quite a mature technology by now in terms of mapping forest structure. And Phil showed an example on how actually now you are using that for functional traits. A few years ago, again, structure from motion is a technique that comes from computer visualization and was developed many, many years ago. Given now that we can take very high resolution images, many of them continuous and overlapping images with these UAVs, now we can create orthomosaics or 3D structures. And this is kind of a cheaper way to do that compared with a more expensive and complex LiDAR. And here's an example that we tested last year. We went to a maple forest. And of course, when you have a photo, that's not going to go through the canopy, different from LiDAR that is going to actually reach the ground in many cases and give you that beautiful cloud point. It's not the case with structure from motion. So we want to create a LiDAR-like scenario. We will need to go into leaf off, leaf on, and then try to match those two layers and have something LiDAR-like. And still we did that and say, well, what is the best we can do if we cannot see the ground, if we don't have ground control points? And we end up with around 1 meter error, 3 centimeters in height compared to the ground measurements. So pretty decent. And again, this was one example or one test we did to test our structure from motion. More important, and again, for peatlands, it works great, and for salt marshes. We did an experiment here with Gail Shamura in the salt marsh, and then we've been mapping peatlands. And when you need to understand the microtopographic elements of a peatland is when the structure from motion and UAV really shines. We have already put a couple of papers on the use for motion in peatlands, and you get what we call now ultra high resolution in those orthomosaic and 3D models. So it's a fantastic tool right now in the application. Hyperspectral remote sensing, and I go just for the sake of time a little bit quick since Phil already covered a little bit of hyperspectral remote sensing. Again, at least that paper from 1988, it made it to New York Times in 1999, so it means that, it was catching the eye. More important, it made it to Netflix. How many of you have seen this movie? Oh, come on. Really? spectral is the scientists that, there is some goals due to a nuclear something in Moldova, and a hyperspectral scientist developed goggles. And actually, they can detect the ghosts in that town with the goggles. And actually, they develop a hyperspectral weapon that they put in a big Hummer. It's spectacular. You have to see that. The science doesn't hold, but at least you will see, something to do this weekend. In all seriousness, what is hyperspectral? In remote sensing, we usually talk about multispectral and hyperspectral. Your RGB camera, it has 3 bands. We see red, green, and blue. A multispectral has 7 to 10 bands. It's your usual Landsat satellite, your Sentinel satellite. So basically, when we have our currency is the spectral response, which is a graph that looks like that, or this, where you have your wavelength, usually from 400 to 2500 nanometers, and you have your reflectance, which is a physical unit of the light that is reflected by the body that the sensor is capturing, using the sunlight as the input energy. Hyperspectral, and that cube was shown previously, it gives you contiguous data, continuous band, very narrow. So actually it creates a full signal that has way more information than a multispectral system. How do we use that? You already saw a full presentation about it, but we can do cool stuff as well. When I was at La Selva as a researcher there, I was like, we have this phenomenon in biology called daiichi. It's pretty much females and male trees in different individuals. And I'm there at La Selva sipping my coffee. It's okay, do female and male leaves reflect different? Do they have different spectral characteristics? And actually, we choose 2 of the main timber species in that area, in the northern zone of Costa Rica, grab a lot of canopy leaves, measure them with a field spectrometer, and compare the actual spectral response, and we found that actually they are different. At the leaf level, males and females of Hieronyma and Virola, that are the 2 species we study, that actually they're different. And we did chemical analysis, we got chlorophyll, nitrogen, and came with a theory of why these are different when the females are producing flowers. So again, very detailed analysis that you can get with hyperspectral. You can do canopy level. This is, again, airborne hyperspectral mission in Costa Rica, led by the Costa Rican government and NASA. They map around 85% of the country with airborne hyperspectral. The HiMap system, I had been collected for part of my life, forest management units with different logging intensity, and I say, can I separate them if I use hyperspectral? And I had very good information on the ground. What you see here is an n-dimensional visualization of the pixels and the reflectance structure from them. And yes, we were able to separate those based on hyperspectral imagery. More important, landscape level. Again, here was, it's funny when you write a paper and you're a co-author and many co-authors. When I was putting this presentation, it's like, oh, Phil was a co-author here too. So I knew Phil by publishing in the same paper, but we met probably last year in a Cabo meeting. So, these things happen. So we use hyperspectral for mapping plantations in Costa Rica. So you have different levels that you can take advantage of the wealth of hyperspectral information. Why do we need a UAV system? Basically, Etienne introduced a little bit of this upscaling process that we are trying to implement in Cabo. At the end, no field data, no airborne data is going to be enough to be able to map those large-scale changes at the global scale. We will need satellite imagery, because satellite imagery is going to cover hundreds kilometers that we need to use. So when, for example, ESA, the European Space Agency, put Sentinel-2 satellite 3 or 4 years ago, they needed a validation. And we used a Peatland, the Merblue Peatland, Ray Software, which is here, led that project. And basically, in collaboration with Magil, we went and did field spectroscopy, and we did airborne in order to validate Sentinel-2 products like NDVI, reflectance, and trying to understand phenological changes in mere blue using those products. What we understood is that, again, going to Etienne's, very costly, you cover a little area, a little bit expensive, you cover a large area, but sometimes the number of samples you get here are not enough to actually make the link, the relationships that you need. And I don't know if that is the case, Phil, in your areas where you have this gigantic area, and then you have only little number of points that you can correlate, and then your relationships kind of mess with your thinking about the ecosystem. I call this the middleman, the UAB. If we have a UAB platform that can expand our field ground measurements and they are representative of our field measurements, then we can have more points to basically complement this and validate the larger scale. What are the challenges? Remote sensing hyperspectral UAV has been around for the last, I would say, 9-10 years. And some of the most common system is what we call a push broom. Basically, there is a line imager. Every line is collected in the forward direction of your platform. And basically, every line or every image is going to have your spectral. Every pixel that is collected in that across track is going to be your reflectance. When you have a full image, you have your spectral data. We understand fairly well or very well by now the airborne platforms. I mean, the GPL, NASA have been doing this for many, many years. NRC has been doing this for many, many years. And we have a very good understanding of how to collect this data, how to analyze it, how to correct it. The problem is that when you move that to an UAV, now you are collecting a centimeter resolution. So any vibration, any attitude movement of your platform is going to be shown here. And just to show that, these are one of the first papers published in a hyperspectral line imager. Here is by Lucier 2014. And I don't know if you can see on the back, this is the non-geo-corrected data. This is the geo-corrected, and you have a lot of non-data points there, or lines. Not only that, they have fixed points to see the geo-correction, and you see the errors between the points and your ground data. And it's because using UAV hyperspectral is not easy. And that's why it took us around 2 years to develop and be confident that the system can be actually implemented as a research tool. And I will talk a little bit more about that later. So our system is the MicroCasi. It's a Canadian company, it's ITRIS. They produce the ones, also the instruments for the airborne, which are the CASI and the SASI that we've been using, or NRC has been using for the last 10 years. The spectral range is from 0.4 to 1 micrometer, 280 area channels, so it gives you that beautiful curve that we all love in hyperspectral remote sensing, field of view 34.3, and so on. We can discuss the specifications if you're interested more in this system later on. These are the general system components. Here we have the system finally put together. It's a DJI Matrix 600 Pro plus a DRTK, running a MicroCasi and the custom inertial unit. I will describe very briefly the different components here, and I put the price there so you have an idea of how costly is one of these systems, because they are not cheap. Hopefully in some day they will be more, they will be cheaper. So basically we have the DJI 600 Pro, which allowed us, the maximum payload is 21 kilograms. We have the RTK, which allowed us very precise flying. When you do hyperspectral, you want your lines to be straight. You want minimal movement in the X, Y, and the Z. So this type of system allowed us to do that. And I will show you an example in a few minutes about it. Again, these systems are redundant. So if one of your antennas fails in the craft, still you have two to work with, and it will keep the data. More important, all the data in the craft is recorded. You can extract it after the flight and analyze the quality of your flight. We use a running gimbal to actually stabilize the system, and one of the advantages, again, our UAV pilot or operator is here, is Oliver. Please, Oliver. He has hundreds of hours now in experience. He hasn't crashed only once, not with this system. I will lose my job. He crashed my system, but basically, we spend a lot of time in the stabilization of the system. Again, stabilization, straight lines, low vibration is paramount in getting good data in hyperspectral UAV. We had a custom built both an IMU GPS. We used the standard in the market during the first year of this experiment. It didn't work. It wasn't good for our system. So again, we're an NRC. We have a flight research laboratory. So I went downstairs. I talked to instrumentation people that has tons of experience with IMUs. And I asked them, could you build one for us, please? And they say, sure. Let's do it. So actually went up with a customized IME unit that actually works with the system very well. And again, weight is paramount in a UAV system. So our system is around 7.9 kilograms, which allowed us 10 to 12 minutes of flying time. That is around a flight line about 1.2 kilometers with a width of 27 meters, ground space distance of 3 centimeters, which is pretty good for most of the applications we do. Please work. This is an example of the actual platform flying at MerBlue, our water table experiment for Deep Inandar that is presenting later on today. So that's a happy day for me. It's collecting good data, it's not crashing, it's very stable, and this is basically the system now collecting data at MerBlue. We have to do a lot of tests before being confident of the data quality we have. Here we have Ray Sofer again. His expertise is in calibration validation, so he makes sure that the quality, the spectral quality of the microcast is comparable, that is applicable, and that's a great advantage. So basically, we spent around 4 months testing the system, flying every weekend, flying every week until we were fine-tuning every aspect of the sensor, the platform, and the data acquisition. Let's jump into some examples. This is how our mission planning looks like. This is for Dr. Koop's site in Coalition Garriock Preserve in British Columbia. So basically, we have here our fly lines. We need some geofences to tell the instrument where to start gathering information, when to stop gathering information, because again, this is 280 layers of information at 3 centimeters. Your data is really heavy, and I cannot stress how big are your files once you finish one of these missions. This is one of the products that there is your forest, Dr. Koops, at 4 centimeters, and the tree looks nice. Still, you do that the best you can. You still see some, errors here from the data for those that we have a trained eye with imagery. How can we correct that? We need to go back to the drawing board and say, well, try to get the best data possible. And again, this is from the structure from motion. We use both approaches. And basically, we start now analyzing the spectra because that's our currency at the end. We want to see the spectra of the different features there. Not only that, we are moving into data fusion. Can we actually put together your spectral data with your 3D data? So we start thinking about it. Nicholas is one of Margaret's PhD students. He's working with LiDAR and elevation models for a long time. And now, thanks to his algorithms, we start testing how to fuse this data and basically get your 3D hyperspectral. And voila, this is our first example, very fresh from the oven, probably last week. You produced that, Nicholas and Margaret, yeah? Where you have a full 3D hyperspectral point cloud. You have your 3D plus your layer of hyperspectral data. Take-home lessons. Sensor stability is fundamental for hyperspectral at that resolution. At 3 centimeters, you need a stable system. You need 1 meter, doesn't cut it anymore. Trust me. We want a few centimeters. Flight planning is fundamental. You need to understand your system, your ecosystem, and the environmental variables that you are mapping. And yes, right now it takes a lot of people to do this. Right now we were 12 on the field. Some of the colleagues make fun of us. It's like a whole crew there. We look like one rock and roll band in the bus. You know, your operators, your safety at the NSA UAB, there are regulations in every country that you need to fulfill. You need to charge batteries because you want to take advantage of your day. And you want your people in the ground characterizing your atmospheric conditions and all that. Thank you.
It's great to see all the details of the platform that's been used. So just purpose of time and make sure we have enough time to drink our coffee and talk with Pablo.
Yep.
So we won't take any questions.
Yeah, for sure.
We'll leave more time at coffee. So thanks again very much.
Thank you.
We'll just take the microphones.
Oh.
Right, so this is, you're confident it will work? All right, so our next speaker is Isla Myers-Smith. Isla is a Chancellor Fellow at the School of Geosciences at University of Edinburgh. She studies effects of climate change on tundra vegetation. She's an incredibly productive scientist. I just went online. She published, submitted 23 papers this year or something.
I know, it's crazy.
This is 1 every 2 weeks. I don't know if she does it. Maybe she will tell us at the coffee break. And she will oversee research at Cabo side, most northern side in the northern Yukon. And she's really now a world expert on the ecology of the Arctic. Effects of climate change, but particularly the topic of shrub expansion in the Arctic, and I believe that's what she'll be talking about.
So, does anyone know how to switch to the HDMI cable?
Wait, set up the microphone and I'll try to find out.
That's promising.
No signals.
I can do it. It's about a gigabyte. That's really it. Okay. I don't.
Should we switch?
Sure.
Have you figured out?
Yeah. Well, okay.
Okay. Sorry, bit of a technical problem because I think Isla has some really nice video.
I do.
So we will have to switch a gigabyte. It's a very large presentation. So if Crystal doesn't mind, maybe we'll just switch orders. Thank you. All right, so I'll make another.
This really is a spectacular screen, isn't it? I feel like I'm at NASA with a hyperscreen. It's great.
Thanks, Crystal. I'll introduce Crystal. Crystal is a professor at the School of Environment, University of Massachusetts, Boston. Our research, our interests include modeling, reflectance, anisotropy, and albedo. And she uses remote sensing data to monitor vegetation disturbance and phenology. She's assigned science team member for MODIS, collaborator for the NASA Global Ecosystem Dynamics Investigations Space-Based LiDAR mission, which is scheduled to fly on the International Space Station. Right? And she's been an excellent ambassador for Cabo, and Margaret told me at the last conference, she told everyone about it, and more people know about it in the US apparently now than Canada, so that's not the case after the meeting.
Isn't that my job as a collaborator on this? Yeah. Anyways, first of all, it's great to be here. I think this is a very exciting project. I've done a lot of work with Neon as well, like Phil, and so I'm very excited to see the Cabo experiment move forward. Obviously, I put my name up there, but as with any senior professor, really, my graduate students and my postdocs do all the work, and I just like try to rustle up cash and stuff like that. Give the occasional talk. If you do get to come visit us, most of us, UMass Boston was traditionally a commuting school. It's not the flagship. The flagship of the UMass system is out in the western part of the state. But there's been a big push to bring research and development to the Boston campus. And we do have a lovely location right out on Boston Harbor, which is kind of fun. Oh, and how do I move it?
I think this should work.
Okay, thanks. Yeah, there we go. So I have been particularly fortunate in my life to have seen a few satellite launches. With apologies to my kid who was in the audience here, I have to say that watching the satellites go up is one of the most emotional and exciting things I've ever done. Kind of beat out the birth and everything. Terra was a particularly exciting satellite to go up because we had been working on it for 10 or 15 years. It launched right in the last days of 1999, and for the older members of the crew, you remember the entire world was afraid that all the software in the world would die when we passed into the world the year 2000 because no one had thought to make enough space for 4 digits in the year. And so we were all very excited to see it go up, not to mention it was incredibly over budget, so Congress was on our butt. And to watch it finally go up in December of 1999 was spectacular. As you can see, it was a daytime launch. And when you put 10 years, as I say, when you put 10 years of your life into a satellite, watching it actually go up is a pretty emotional experience. MODIS is our workhorse right now in providing imagery of the globe. It goes over the poles every 90 minutes, 14 times a day, and gives us pictures of the Earth almost once a day, twice a day at some points, but it gives us a pretty good view of what's going on on the globe. A few years later, Aqua was launched in an afternoon orbit, so we had a morning satellite and an afternoon satellite. And again, I was very fortunate to watch that one go up. That was a night launch. That was a little bit more like Land of the Living Dead, where all the scientists are walking around going, when's it going to launch? What are we going to be able to see? Not quite as much, but it was still good. The product that I make for NASA is albedo, surface albedo, to drive climate biogeochemical models. And we do this globally. We produce a product from the MODIS, Terra, and Aqua once a day. It's based on surface anisotropy, or the bidirectional reflectance distribution function that we build up from multi-angle observations and give us a real idea of what the surface reflectance is in a whole hemisphere, what the albedo is, what's the percentage of the radiation coming in that gets bounced back out. I like to show these two pictures, even though they're rather old, just because you'll see in January, we can see a little bit of Antarctica. Nice bright Antarctica. We can't see so much up in the northern climes as we're in polar darkness. And you can actually see India. You'll notice when we look in July in the height of the monsoon, we never see India. I must get an email once a month from some poor little researcher in India going, how can I see what the satellite sees in July? And I'm going, nothing. It's cloudy, guys. I've been in India in the monsoon. You can't see up and I can't see down, so you're out of luck. And there's not much we can do about the black spots, the cloudy bits, as the ITCZ takes out the tropics and takes out India and things like that. One of the things I do work a lot on is phenology of both natural and anthropogenic crops and backgrounds. This is a particularly pretty sequence, watching the colors change. This was for a book that we did, so that's why it's particularly pretty. But you can see the swath of the colors changing up here in Canada and then swooping down into New England. Of course, we're somewhat worried in New England as our maples fade away and only move up to Canada and to Quebec, that if you want to see colors, you're going to have to come up here to see them, and get not just New England maple syrup, but Canadian maple syrup. So we do a lot of work on phenology monitoring, especially now that the product is produced each day. We can get a good sense of what's happening on crops and things like that. A few years later, we launched the follow-on for MODIS. This is the NOAA NASA operational satellite, SUMI-NPP. This was, again, a night launch, which, again, is a little bit like you just get to see a bright blast, and that's all you get to see. From that, we also produce broadband white sky albedo. We don't have as much coverage because there's only one SUMI-NPP up there. Now there's been a second one launched, but we don't have a morning and afternoon. They're always running at the same time, so we don't have quite the capability we had with MODIS, where we had a morning bird and afternoon bird. But we're getting some really good results from this too, so that gives us... MODIS is getting kind of old. It was 1999, so MODIS is getting ancient. And so the VIIRS sensors on SUMI-NPP and the JPass satellites that are coming along will have to pick up the work and carry on. One of the hardest things about having a global product is you're supposed to, in theory, validate it, but how on earth do you do that? We're pathetically grateful for networks like NEON, like Cabo, like FluxNet, baseline surface radiation network, things like that, where we rely on the data that are collected on the field. Since I do albedo, I rely a lot on tower-mounted albedometers. Here's an example at Desert Rock, which is a BSRN site, a SURFRAD site, and Harvard Forest, which is a NEON site. Harvard Forest is the closest one to us in Boston, of course, and so we're quite relieved where we may miss a few points. This is when it snows, and if we can get some snow that's just around the tower, but at a 500 meter MODIS pixel, it may not look like snow at all, so we may miss that. So you always have to take, as the previous speakers have said, you have to take into account what your validation data may, even at a tower, it may be 100 meters, but that isn't all that great, that isn't all that spatially representative of maybe your 500 meter or 1 kilometer footprint. So we spend a lot of time worrying about that. Whoops, wrong way. There we go. Landsat. Landsat's our granddaddy of satellites. I just made a note here that in July 1972 was the first Landsat 1, and Landsat 8 went up in 2013. We're on tap to get Landsat 9 up in 2020. It was a little bit worrisome. The American administration was fairly anti-Landsat for a while, but we seem to be hanging in there even under this administration, that the next Landsat will move forward. As I say, I was fortunate enough to see this launch too, which was quite exciting, and another daytime launch. Close up there of Landsat 8 soon after it launched of the Boston area. So for you guys, very excited about these sites. They're very interesting sites. And I had a really good time going into our North American albedo map that's made from Landsat and kind of messing around and saying, oh, okay, good. Well, glad I'm making albedo over all these spots. That's lucky. And we're funded to do a North American albedo as part of the Landsat team. So we'll build up a time series of these. So that we can provide that sort of information back to you, and perhaps use some of the information that you're collecting with your pyranometer and stuff to help with validation as well. I'm always pathetically grateful for any validation data. I do want to mention that, and Pablo I think mentioned that the Sentinels went up. Sentinel A in 2015, Sentinel B in 2017. The European Space Agency sensors. They're fabulous. They're like Landsat-like instruments, but even finer resolution, a few extra spectral channels. They're just great to work with. We're so excited. And what we're particularly excited about is if you couple together the Sentinel 2A, 2B with Landsat 8, you get really good coverage. One of the problems with Landsat is you only get a picture every 8 days, and that's if it's not cloudy. So that means, like, particularly in the shoulder seasons, the phenology, when you're most interested, often it's too cloudy, you can't see anything. So we're very excited to get these frequent looks, moving things up to every 5 or lower days. We're starting to get looks, so we have more of a chance we'll get a cloud-free look. Particularly as we move up to the higher latitudes, I particularly want to talk to Islip because we are funded to work with the harmonized Sentinel 2AB Landsat data, which has just been released in the last week. The first initial attempts at it, where they're putting together all the data. NASA's funding this project to put together all the data, try to make all the atmospheric corrections, the channels more consistent. So it's a corrected data set that we can work with. And we're focused on trying to come up with a high northern latitude albedo north of 40 degrees. I picked 40 because I wanted to get Harvard Forest in there. But we're going to be really focusing at these tundra and boreal and tundra ecosystems. So we're quite excited about that. The other thing that I do is a lot of LiDAR, a lot of terrestrial LiDAR. We have in the past played with some of the big fancy LiDARs, some of the research LiDARs. Nicholas and I have worked on the echidna instrument from Australia, which, by the way, the echidna is actually an animal, but it's a spiny anteater, hedgehoggy-like looking thing from Australia, and it was named that because they thought all the beams going out from the LiDAR looked like the spines of the echidna. When I was last in Australia, I took the whole team to the zoo because I was like, you're going to see a live echidna if it kills me.
Well, they won't kill you.
Huh?
They won't kill you.
No, they're very sweet. They're very sweet. But more recently, so we worked with the high-powered guys, and they're great, but they take a really long time to collect data. You're sitting there in the forest going chug, 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 boom, 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 boom. And you're kind of doing the LiDAR dance. You're always kind of standing behind the thing and moving around. And they're great, but they take a long time. So we came up with this one, which is, and there's some commercial ones that are coming out now too. These little portable guys that are just great. They're not as strong. They don't go as far. They only go 35 or 40 meters, but they're cheap, they're fast. This one works in 33 seconds to do a complete scan, so we can cover a lot of forest and wander around. This is just a picture of the compact biomass LiDAR, perched it, photoshopped into its scan. And they're not as high resolution, but you pretty much know what you're looking at. It's called the compact biomass LiDAR. But it's the CBL, but really its name is the cute baby LiDAR. The very first one was ever created by Jan van Aert up at RIT out of parts out of his lab. And when I saw it at a ForestSat a few years back, I was squeaking at him happily about, oh my God, it's so adorable, it's so cute, Jan, can I have one? And he said, well, no, but I used up all the parts, but if you've got a good engineer, you can build one. We could tell you, help you build one yourself. It's based on a sick commercial LiDAR that's mostly used on assembly lines to make sure little Tommy doesn't stick his hands in the works and stop the assembly line. So anyways, by the end of the conference, everyone was calling it Jan's cute baby LiDAR. And then Crystal made another one, and in print we call it the compact biomass LiDAR, but if you ever see CBL, it really means cute baby LiDAR. Leica has a new one out that's around $20,000. We've got one, we're playing with it, it's quite nice, a little better resolution. So these tinier terrestrial LiDARs are really taken, and they're being driven by the UAV because they're trying to get LiDARs that'll work up on the UAVs and are light and small enough. As I say, we mostly work at Harvard Forest, which is right up there where the symbol is. I just spent a year sabbatical out at Harvard Forest, which was a blast. I had the most adorable cottage out there. I kept saying I felt like Henry David Thoreau. I had gone to the woods to live deliberately and fully experience the forest. And it's up there by Quabbin Lake, which is the reservoir for Boston. Landsat image of the area. And one of the things that also makes Harbor Forest kind of interesting is, very sadly, we're on the forefront of the hemlock woolly adelgid infestation in New England. Hemlock woolly adelgids have been marching up the coast of the U.S. since 1950. They're pretty much killing all of our eastern hemlocks. Looks like it will lose every single one of them, just like we lost the American chestnut. We so far don't have a, you can fill them full of chemicals and keep an individual one here and there going, but we're not really finding ones that can fight their way against these little insects. They basically build a little cotton nest egg sac at the base of each and every single little hemlock needle. So there's gazillions of them. And one of our biggest fears is we walk around the forest that as foresters, we're spreading them throughout the forest, but of course birds are doing that too. And I'm sorry, but it's crossing into Canada. It's up through Maine, despite the U.S.'s best attempts to keep wood products, hemlock products from moving north. And the real reason why it's moving north is because of climate change. The only thing that kills them is extensive cold. Now, fortunately, it's cold today, but we just don't have that kind of cold like we used to down in New England, so they're heading north. But it's very interesting from a LiDAR standpoint because it's all to do with structure. It defoliates the bottom branches, the bottom needles. The top may still look okay, and as you fly overhead with UAV or aircraft, it looks sort of okay. But when you're messing around underneath, you're seeing, oh, they're all dead and dying. It's a little bit like being at the end of the Lord of the Rings movies where all the leaves are coming down on your head. It's very depressing. But what is coming up now is understory growth at Harvard Forest. It's all black birch. We've got a new deciduous crop coming up, but it's changing the whole ecosystem. Hemlocks kept things moist, cool, provided habitat, helped with our stream runoff and things like that. So it's a pretty extensive ecosystem change that we're dealing with right now. And but we're having, on the plus side, we're having a good time running around in the forest with our terrestrial LiDARs documenting it. Whenever we document we try to come up with biomass estimates of what's happening. Now the community of terrestrial laser scanner people are all pretty much using various models that try to turn the point clouds in the middle into solid objects. A lot of us are using Pasi Raman's quantitative structure models where he fits a bunch of cylinders starts at the base and chugs up the tree and turns all the branches and little bits and pieces. That's from the CBL. Again, we're not a very high resolution thing. If you've got a Regal or a fancier instrument, then you can make it out into the finer twigs, but it still does a pretty good job in giving you the rough estimate of the biomass. That's what's going on. The other thing that's great about Harvard is it is a NEON site. We've been very fortunate. We've had several NEON overflights already. We also routinely convince NASA's G-Light instrument into overflying, more as a favor. G-Light got a LiDAR, a hyperspectral instrument on it, RGB imager as well, and a thermal imager on it. Now, they actually have 2 discrete LiDARs on it right now, so they're getting a lot of hits per meter. Very, very exciting looking at those data. NEON, of course, is a full waveform LiDAR, and the hyperspectral. And as I say, we've had several overpasses. They overflew this summer. They've overflown several times. So we have a really nice airborne component of before the hemlock woolly adelgid through to now after the hemlock woolly adelgid. So that's one of my graduate students is actually working frequently down at NASA on that work. Traditionally airborne laser scanning, and that's true for ALS, have given us canopy height models, a really nice job, LiDAR, or whether it could be structure by motion. It's doing a really nice job of what your bumpy top of canopy looks like. If you have the terrestrial LiDAR underneath, then you can start building the bottom up. What's the bottom of the canopy? What's the understory? And you can couple them together and get things like canopy thickness and a little better rendition of what the ground looks like. This is an example where you've got a G-Light overflight, lots of pixels, but it's just, you can see that's the top of the canopy. But you don't know what's going on underneath. You put in the terrestrial LiDAR, this is CBL, and suddenly you can build all of your understory and canopy structure. which is really exciting. Oops, the wrong way. And you can start linking this up. So that's what you're getting from neon, that's what you're getting from G-Light. And then down here is what you're getting from more of the CBL. And from the ground, you're getting, some of all of them. Maybe a little better from the terrestrial scanning LiDAR, but you're getting something from all of them. We were fortunate. I started my sabbatical out at Harvard Forest with it's like you guys running this thing. It's always a lot of work. We ran this international LiDAR scanning and destructive sampling campaign. My mentor, my dissertation advisor, Alan Strahler at BU, kept inviting people. And I had to keep saying, Alan, you've got 57 people on the ground, and we're working on a 50 meter by 50 meter square with 9 LiDARs. How the devil are we going to do this? You in theory taught me to do field work. Well, you stop inviting people. So we had a lot of people, Harvard Forest Woods crew, Virginia Tech, UMaine, Michigan State all sent foresters, which was a real privilege to work with foresters that did the destructive sampling, did all of the weighing, the drying, the wood density, tree rings, blah, blah, blah. Harvard Forest lent us a bucket lift. I kept saying, great, we've got all these participants, all these LiDARs, a bucket lift, tarps, chainsaws, what could possibly go wrong? And actually we did pretty well. We also did a leaf off and a leaf on, since it is a primarily deciduous and hemlock mixed spot that we were working on. There's one of the sensors on the ground, and Bucky, the bucket lift. People were scanning, at least we could scan with the CBL. Some of the fancier ones, the vibration up on the thing is too much, but we can get away with a little bit coarser one to get a top of a particular tree like that. And then we can shove them together. We can have the purple from the ground. We can have the top in green. We can piece the two together. And this is actually kind of humorous too. It even catches the operator up in the bucket and floating around up. We have to take him out. It messes up our biomass measures. And then, as I say, watching the field crew take these huge trees down was really impressive. Core, everything's been shipped. We couldn't ship any hemlock. We could only ship hemlock south because Maine still has a restriction, even though it's already in Maine. But so it was quite a complicated project. I'm just going to end on one more experiment, which is some work we've done at Plum Island, mostly because of Boucherville. And this is a long-term ecological research site. Harvard, by the way, is an LTR too. It's as well as a neon site and a flux site. Plum Island, they're just north of Boston. We've convinced G-Light to overfly a few times, and you get great imagery of the salt marsh. You can see something of what's going on the platforms leading down to the creeks. We deployed the CBL as well over 3 summers to try to get a really good 3D model of what the platforms were, where the different kinds of salt marsh components along the way. One nice thing about having a cheap LiDAR is you usually don't mind risking your graduate students in the muck, but we also don't mind missing your terrestrial laser scanner because it's cheap. So we do stupid things like run it on a zip line between 2 sticks, basically, and scan the creek that way rather than go out with measuring and trying to build up the creek bed that way. And we could get this kind of information that we could embed over the airborne, which is kind of fun. So ending on this note, as you mentioned, I'm also a collaborator on this instrument. We've been trying to get this LiDAR up in the air for 15, 20 years. At the moment, the launch date is December 4th or 5th. I'm actually waiting to see if that's still solid. We're a little bit worried because we were supposed to get our invites to the launch last week, and NASA hasn't spoken to us, and we're all going, guys, is it going? Are we going? It's a space-based LiDAR. It was supposed to be a standalone. The best we could do was to get it up on the International Space Station, which means it's not going to cover Canada much. It's only going to go to 54 degrees, which is extremely disappointing. But ISAT-2 launched. It's not a full waveform LiDAR, but at least it gives us kind of the top of the canopy. And I've seen that FORSAT, they showed the first pictures from ISAT-2, so at least we have a top of canopy from ISAT-2. And then we're a little bit lower in latitude. We do have some ability coming up from Jedi. Jedi he's really a Star Wars fan. You know, we celebrate May the 4th be with you and all that good stuff. So we're very excited about this. It's going to be about a 27 to 30 meter resolution for each pulse. They'll be producing some of the data on 500 meter gridded cells, which isn't quite as exciting. But we're really excited about this as a new step moving forward in LiDAR. And we're really excited about Cabo and having all this UAV. Now, one thing about phenology is when I fly these aircraft, I'm really lucky if I get one overflight from Neon every year, or maybe every other year. I get one flight from G-Light. But with the UAVs, you can fly a bunch of times, particularly over those shoulder seasons when things are greening up, when they're senescing in the fall. So it's really, really exciting. And that's it.
Thank you very much. Thanks, Crystal. So while we load Isla's presentation, we probably have time for a couple of questions for Crystal.
I really want to work with you.
Any questions? No? All right. We'll keep to.
I'll go ahead and ask. This is just a totally, since I haven't worked with the terrestrial LiDAR scanning, I'm definitely a follower, not a leader when it comes to that, but very interested in that. And the reason why I avoided using them is, I think you hinted at this, is that the availability and the processing of the data. I don't want to be the developer of the methods to process the data. How mature would you say the methods for working with the terrestrial scanning?
So I took the slide out because I knew I was over anyways, but we have a terrestrial laser scanning interest group that's been running for about 7 or 8 years now, that's also supported by an MSF RCN, or Research Coordination Network. And so that team of us, the 30 of us that are really working on terrestrial life, we're all swapping stuff all the time. So like I say, we're using several of these models that turn point clouds into so it's definitely maturing. It's getting easier and easier. The commercial sensors are supplying software that's supposed to make it really easy. The trick is they supply that, and then you have to go in and still tinker with it to get the point clouds to snap together. If you take 3 around the tree, it sort of works, but then you got to image them a little bit yourself. We use Cloud Compare, it's open source, to do that imaging. Yep. So, I think we're getting there. We're reaching a consensus, and that's one of the things like this field campaign is trying to do, is bring the foresters that came to do constructive sampling in with us so that they can start using this and their students can start using it. So we're definitely moving forward with that. And the UAV LiDARs are great. They just have tended to be kind of expensive. The top of the line, the Regal Bucks, there's only 3 or 4 of us that have paid $500,000 to do one of those. So we're just starting to see that data now. So most of us have been happy to have, airborne, try to match airborne with terrestrial. But we're looking forward to getting more LiDAR so we can do it more frequently. This is not that hard. 33 seconds to scan. One student can go out and in the morning knock off, 30 by 40 hex area pretty fast, and then we got something to compare to.
Thank you very much, Crystal. Timing is good because I see the coffee and snacks and things have arrived. So our last speaker is Isla. I've already introduced Isla, so we'll just jump straight in.
Launch right in if this works. So I think maybe that was a nice order there because you set up some excitement about what's going on up north. So you folks all know that a huge portion of Canada is the Arctic, and what's really exciting about this Cabo project is to embrace that. So I'm going to tell you a little bit about some of the biodiversity and greening changes that are going on in the high latitudes of the planet. and a little bit of the work that we're doing in the Canadian Arctic. So this is one of the Cabo field sites. This is Kikiktarik Herschel Island. It's off the Arctic coast of the Yukon Territory, so sitting just next to the border of Alaska, quite close to the town of Inuvik, but you do need to charter a plane to get there. And this is a Felis Navarre's John sitting on the edge of a retrogressive thaw slump, so a huge permafrost feature. So that is a sort of cliff of ice there. It's about 40 meters tall, and that feature is regressing back into the landscape at about 15 meters or so a year, and it's about 2 kilometers across at the moment. So this is an area where these landscapes are changing very rapidly, and that's influencing the plants and the biodiversity on the terrestrial part of this landscape. So one of the sort of big questions, of course, we all have is, how is the Arctic changing? And of course, the first thing we think about is warming, and we often think about these kind of iconic photographs. So this is actually a student of Mark's and a former postdoc of mine, Anne Bjorkman's photograph, not the polar bear. Yeah, she took this photo from a helicopter up near Ellesmere Island. And so 2 polar bears here on some melting ice. And so this is the sort of iconic image that we have of the changing Arctic. But I like to sort of change the focus a bit to these fluffy creatures. They're white, they're cute, they're just like a polar bear, but this is Eriophrum vaginatum, and it's one of the dominant sedge species in the Western Arctic. So the Arctic is warming. You've probably seen these circumpolar maps, and I want to sort of shift the focus away from the southern regions and looking top down on the planet. And so all the red areas there are where there have been substantial warming over the last 30 years or so. And it's about 2 degrees of warming over the last 3 decades in the Arctic. And another change that's been going on is a sort of greening of these Arctic systems, which ties nicely into the last presentation there. So again, this is that retrogressive thaw slump seen from a drone. And then we can go all the way up to space here. And so if you look across some of the longer term satellite platforms, you see this sort of strong greening pattern at high latitudes in many of the pixels, with associated browning in a lot of the boreal forest regions. So one of the questions that is posed by these kind of satellite trends is whether this greening is really due to vegetation change on the ground. So perhaps climate warming is leading to increased plant growth, and that could be one of a variety of factors. So it could be earlier green up and flowering of the plants in these systems. It could be changes in plant cover and the composition of these communities, and it could be changes in the traits of those communities, like, for example, increases in canopy height. And all of that change really matters because in these high latitude systems, and so the link to albedo there, vegetation change can influence climate change through large scale climate feedbacks. So either through the reflectance of the surface, through heat being trapped in the system and thawing permafrost, or through changes in nutrient cycling and carbon storage. Vegetation change in these high latitudes can influence the global climate system. And so one of the ways to get at understanding how vegetation is changing in these high latitude systems is actually by compiling the data sets that we have on the ground in situ at sites around the Arctic. And so my research group and my collaborators have been working for the past, decade or so at trying to compile all the different data sets that can help us inform our understanding of what changes are going on in these high latitude systems. So I'll talk about some of these today, others I won't. But in particular, the International Tundra Experiment Network, which has been running for over 30 years, compiling data on community composition change at sites around the Arctic. So I'll pose a few questions about what types of plant changes might be going on that might help explain that greening. And we'll start with the question of how tundra phenology is changing. So this is a phenocam, so a time-lapse camera setup at our Kikiktarik Herschel Island site, and you can see the plants flowering and greening, changing, and then eventually leading towards senescence at this site. And you can also look at in situ records in plots and along transects of that greening and senescing of these plants across the growing season. And so this is a plot of change in leaf phenology for one species, Salix arctica, over time from the early 2000s through the present. And what you can see at the Kikiktarik site is that we're getting an earlier growing season, and the senescence is also occurring a little bit earlier. So the growing season length is not particularly changing over time, but everything's shifting a bit earlier. And these growing seasons are very short. So you can see in the photographs on the far side there, you've got pretty brown tundra landscape in the middle of June, then things rapidly green up, and then you've got senescence fully underway by mid-August. And so this rate of change is about 5 days earlier per decade. And if you look at the phenological studies for, say, forest species in temperate North America, the amount of change that they're seeing is only on the order of, 1 day or less per decade. So these are really rapid rates of change. And when we look across the International Tundra Experiment Network and the phenology data that has been collected across those sites, and we actually see examples of green up getting earlier, but also examples of green up getting later across these sites. And if you look at where there are existing data and you look at the distribution of that change, it actually sits nicely on 0. So we're not seeing that earlier green up occurring at all the places where we have in situ records. And one of the factors that might help explain that is that snowmelt dynamics differ across the Arctic. So you have certain sites where there's actually greater snowfall and the snow is not actually leaving those systems earlier. And that's not the case at our Kikiktarik site. So another question that we've been trying to delve into over the last while is whether the Arctic is getting shrubbier. So we've moved up beyond the tree lines. We can't show beautiful LiDAR of trees anymore, but we could potentially, if we got a LiDAR system up here, capture these shrubs. So this is, again, for the Kikiktarik site. These are some willow species, and they are about, at the moment, the tallest individuals at this site are about waist high, let's say. So these are the biggest plants in this tundra landscape, so they're kind of the trees of the tundra. And at many sites around the tundra biome, we're seeing an increase in the shrub species. And this is just to visualize what that looks like. So this is the Kikiktarik site again. This is a photograph from 1987. And then when I started working at the site in 2009, and then I'll flip through photographs of the intervening years, bringing us right up to the present day. And you can see that these willow shrubs have invaded this creek bed here and become very dominant in this part of the island. And if we look at the literature and what's been published out there, we see many, many studies that are showing this increasing cover of shrub species coming from a variety of different lines of evidence. So historical ecology like repeat photography, ecological monitoring on the ground, phenology records, dendroecology records, and experiments. And there are other studies out there that show decreasing cover and some evidence of stable cover at some sites, but the predominant signal is for increasing shrubs. And then, so the shrubs are increasing. Is that leading to a change in the community composition as a whole? And so, again, with this International Tundra Experiment Network, we have researchers like myself and my PhD student Gergana there monitoring plots, usually a meter squared, at sites all around the tundra biome. That's us this summer. And so we can look at the change in those plots over time. That very video that you were looking at there, these are the data. So again, starting in 1999 and moving through the present day. And this is what the data looked like when I started working at the site. So sort of plant cover and bare ground, the two plots here. And then I collected the first data in 2009, and the relationships look like this. And I first got worried that I wasn't carrying out the protocols correctly or something. But since then, the trends that we started to see in 2009 have continued. So relatively dramatic increases in plant cover across two different plant communities, and then a pretty strong decrease in bare ground in this system. And if we look again across all the different International Tundra Experiment sites, we are seeing that pattern at many of these sites. So it is a biome-wide signal. So those are sort of observational lines of evidence for how the tundra is changing, but what happens when you experimentally warm things up? And that's what the International Tundra Experiment set out to do by installing these open-top chambers at sites around the Arctic. So that's Ellesmere Island, where I and my colleague Jeff at the back there were hanging out this summer with Greg Henry from UBC. And we were visiting his long-term plots with these experimental treatments and trying to understand the landscape context to the change that's being observed in those warming plots and their controls. And so indeed, plants do grow more when you warm them up. So this is some drone imagery of one of the warming plots with its chamber removed, and you can see the sort of biomass is greater in there. Of course, people have been walking around this plot, so that's, but we can compare those data to the control plots that have been not disturbed as well. And you can see that is also picked up in the spectral signatures. So these are multispectral data collected from a drone. And we also set up a thermal camera over one of these chambers, and so you can actually see that the warming experiment is working. Heat is being trapped inside these open-top chambers across the day. So we're working with analyzing those data to sort of put together the landscape context to those experimental treatments. But we also want to understand how biodiversity is changing across this landscape beyond the plots where the ecological monitoring has occurred. So this is back to the Kikik-Tarik site. And I'll start this time with the tundra biome as a whole. We're not seeing dramatic changes in species richness across the tundra biome. That's not necessarily surprising. These are species poor environments. So even if you got one new species in a plot over 30 years, it doesn't lead to a dramatic effect size. So these are, again, across all the ITEX sites. We see that sort of slightly positive distribution here, but the credible interval does not overlap 0, so we're not seeing richness change across all the sites. And we're also not seeing functional diversity change across these sites either at that biome scale. But we are seeing changes in some of the plant traits in these systems. And so the one trait that seems to be changing the most over time is plant height. And what we're finding about plant height, that it is new species entering into the plots with stronger height relationships that are creating that increase in plant height for the plant communities across the tundra biome. And it's not the species that are lost from the plots that are controlling that change in community level plant height over time. And this, from the Kikik-Tarik site, is one of the species that has invaded the long-term monitoring plots that's increasing to the plant height at the community level at that site. So the question is, where do these new species come from? So they may not be invasive species coming from way far south. They might be there in the landscape already. And so what we set out to do over the last couple summers is to understand where new species that may invade the plots in future are located within the landscape. And these are some data hot off the press. So we have the 2 vegetation communities on the island, and we walked in concentric circles for many hours to figure out where all the new species that aren't currently located in the plots are located within that landscape. And we've calculated species area curves and found that there are a variety of species that are very close to the long-term monitoring plots at the moment, but not currently located within them. So these 4 species are all within 5 meters of the monitoring plots. And within 100 meters, we have between 13 and 26 potential new species that could invade the plots in this landscape. And so the next challenge is to ask whether these species are located in particular microclimates that can help us understand how, as these tundra ecosystems warm, the biodiversity might change. And so that brings us to sort of some of the Cabo goals, which is tying biodiversity change with spectral diversity. And at this site, we're working mostly with multispectral data, but we're keen to delve into the hyperspectral world when that's possible. And we've also been trying to work cross-site around the Arctic. And so Jeff and I established a couple years ago a high-latitude drone ecology network. So the idea being to get people who are already taking drones into systems around the tundra biome to use common protocols and collect data that can then be synthesized, hopefully this year. And so the idea is here to collect multispectral data at the landscape scale so we can start to understand how vegetation communities are changing and how that links to the greening patterns that we're seeing at those tundra biome scales. And one of the questions we have is, so they across the sort of maps that I showed you at the very beginning of the talk, we have these pixels that indicate greening, and then we have other pixels that indicate no trend in a vegetation index, like the normalized difference vegetation index. And could it be that something about the heterogeneity of those different sites or some landscape level parameter could help explain why on Kikik-Tarik we see strong greening, but say in Kangerlussuaq in Greenland we don't? And so we're currently working on these analyses right now, and we've delved into using the Earth engine to compare drone data and satellite data across these different sites and ask what covariates best explain when they don't agree. So that work is ongoing, and what we're excited about in the future with the Cabo project is moving into the hyperspectral realm and asking whether that helps us understand these biodiversity changes that may or may not be going on in Arctic systems. So these are a few pictures from our preliminary Cabo fieldwork this summer. So this is us ready to go collect some multispectral and hyperspectral data this summer, looking very positive and enthusiastic. And then that's what happens once we got out to the site. The fog rolled in and we spent 12 hours waiting for it to leave. And it was beautifully sunny just above that cloud bank. But those are the challenges of Arctic fieldwork. But we did get a little bit of data collected. So these are the hyperspectral signatures from a land-based system for each of the long-term monitoring plots in the 2 different vegetation communities where the ecological monitoring has occurred. And we haven't, these are sort of very hot off the press. I was sent them a couple days ago, haven't really analyzed the data, but it's interesting to see the diversity. So these are all meter squared plots at that landscape scale between these 2 different vegetation communities. So more on this to come. So where will the Cabo project take us? There's the road ahead. I hope to exciting hyperspectral futures. And just to sort of summarize, so there's this strong greening that's going on in the Arctic that we think is being driven in part by warming, and it's probably related to changing plant phenology, changing composition of species, and changing traits of those species in these tundra ecosystems. And all of that may be influencing biodiversity and its positioning across the landscape. And we hope that spectral data can help us understand and scale individual sort of in situ observations up to landscape and biome scales. So I hope I've given you something to mull over here, chew away on, and maybe the snacks have arrived. So I'll move over there. And if you have any more questions, you can check out our website and come chat to me afterwards. And thanks very much.
Thank you very much, Isla. With all the technical difficulties, we're actually not too bad, but we're about 10 minutes late. I don't want to cut short the coffee break. It will be a good time. We will cut short lunch. So we have planned 25 minutes for a coffee break, and I think we should stick to that. But we can take 1 or 2 questions for Isla before we move to the coffee. Yes?
Can you say something about low elevation in terms of greening? Say changes that are occurring at 0 meters as opposed to 100 meters above sea level, 500 meters?
Yeah, so in the Arctic, elevational gradients are also climate gradients. So as you move up mountains within these high latitudes, you're moving into more extreme environments. So the sort of biomass that's on the landscape is decreasing across those climate gradients. At our Kikitark site, it's a relatively low elevational site. So we are seeing vegetation change with elevation across that. And one of the things we're excited about doing with the drone data is sort of linking in elevational topographic data. So the Arctic DM has been recently released. I don't know if folks are familiar with that. So we are hoping to be able to use the sort of DMs we can produce off of our drone data and then to understand how that might influence drone level, say, NDVI patterns, and then scale that up to satellite data using the Arctic DM. So to sort of link across those scales.
Maybe one last question for Isla? Yeah.
Okay. You said that, okay, the plants are getting taller in the one site, and in Greenland they weren't, and you weren't sure why. But when you put up the map of the temperature changes, there's a strip there through Greenland that said it was actually getting colder.
So there's a place, sort of a region between northern Quebec, Labrador, and Greenland that hasn't been warming as rapidly, or that it's actually been warming quite rapidly, but it's more recent, the warming that's been going on there. And that's due to sort of sea ice and ocean dynamics. The data that I showed was actually greening, not plant height. So it definitely can be that you get these sort of heterogeneous relationships between the amount of climate change that's going on and how plants are responding. Some of the things that I didn't talk about in this presentation would be sort of sea ice dynamics, snow melt and precipitation dynamics that are all at play in these systems as well, influencing the greening patterns and how the plants are responding on the landscape.
Thanks, Isla. Right, so just to clarify on your order, we'll shift everything all the way to lunch by 10 minutes, so which means that we'll start the presentation at 10:55.